All right. So I would also like to start by thanking Virginie and Abdou, of course, for organizing this wonderful conference and for having me here. So thank you very much, Virginie. It's good to see you again. And well, one of the few advantages of speaking among the very last speakers that I won't need a very long introduction. Actually, I won't introduce any of the concepts I will present because they've been introduced by other speakers before me, so it will be simple. We've heard a lot about pigmentation pattern diversification, mostly this morning in butterflies, and just before with Trisha flies. I mostly work with flies, but I will talk to you about a project, a side project that we started in the lab a few years ago that has to do with the pigmentation evolution in beetles, and in particular in the coccinellid Harmonia axyridis. And what got me interested in this system is that, well, doesn't hold. It's okay. Well, is that there is a crazy diversity when it comes to pigmentation pattern in this Harmonia axyridis species that have been studied actually from the very beginning of the 21st century by pioneers in genetics, and you'll see their name in the coming slide. And what you can see on this panel is just a subset of the diversity of coloration pattern you can find in the wild in this species. But in fact, there are only 4 colomorphs, which are shown here, that represent about 99% of the diversity you find in natural population, meaning that most of these color forms here are extremely rare, although they exist. So these color forms, as you can see, they come in 2 basic colors, red and black. And you can see, for instance, this what we call the red morph. Basically, it has a red background, red-orange background with about 18 dark dots on the elytra. And those black or melanic morphs have a black background coloration with varying numbers of red dots, 2, 4, and up to 12. And I was very much interested into this pattern of variation for multiple reasons. One is that, as I've shown you, there is an extensive diversity of coloration pattern that is parallel with variation in geographical and seasonal distribution. And this has been observed already by Dobzhansky more than a century ago, for instance. There is also quite of knowledge about the genetics underlying this diversification, and it's been shown that all this diversity maps to a single autosomal locus that contains multiple alleles. It's called the H locus, that have been identified or actually named in the 1950s, but of course not characterized molecularly. And also, more recently, it's been shown that contributing to the diversification of pigmentation pattern in this species, there is also some developmental plasticity that is linked to, in part, to temperature variation. And during this talk, I would like to address those two points. What is the identity of this H locus that is responsible for color pattern changes between morphs, and what is the underlying basis of developmental plasticity? So part of it is published, so I'll go quickly, and the second part is really much an ongoing work, so I will show you mostly unpublished and preliminary data. So to do this work, I collaborated with Mathieu Gauthier and Arnaud Estou, who are population geneticists. So they know what they're doing in this field, more than me actually. They are from Montpellier, not far from Marseille, where I'm working. And we decided to, or they decided, to be honest, to use a mapping approach that relies on the diversification of different populations that are geographically distributed, contrasting basically red populations, or population containing only red individuals, with population containing a variety of morphs, but all belonging to the dark, to the melanic forms. And what we know is that, as I said, that the H locus is autosomal, and we know that there is a complicated recessive dominance relationship, but that makes that the red individuals are homozygous for the red allele. That was known from the 1950s. So they performed a genome-wide association study after assembling the whole genome, and this is the map we got, or the map they got, basically. These are the different contexts we got from back then, and you can see the various peaks, and including this very big, this big tower here of SNPs. And when we zoom in into this region, what we found is that all these SNPs, basically, this huge tower maps to this interval of about 180 kb that spans the beginning of a gene named Panier, which codes for transcription factors in Drosophila, melanogaster, and other insects, and the 5' region of that gene / the 3' region of the upstream gene GATA-E. So we had two obvious candidate genes for which we wanted to test their function. So for this, we turned to very simple RNAi or RNA interference methods that works extremely well in Harmonia, which is you synthesize the double-stranded RNA for the gene of interest, and you inject those double-stranded RNA preparation into the larvae, into the cavity, or even in the pupae. And that's what we did for the Panier gene, and this is the result we got. As you can see, in all the different morphs, you basically eliminate entirely the production of black pigments throughout the body. So the elytra and of course the rest of the body appears completely red. So this reveals that the Panier gene is absolutely essential for the production of black pigment in Harmonia axyridis. Now we wanted to know what is the possible changes at this gene that might be responsible for the divergence in coloration pattern. Well, we didn't see any changes in the amino acid sequence predicted from the genome sequence from these different morphs. So we look into expression data. So we first look at RNA-seq data on the developing elytra, comparing the one red morph and this, one black morph and this red morph. And as you can see, there is a difference in overall expression level of the Panier transcript between those two morphs. To go into more details and to look for potential differences in the distribution, we raise an antibody against the Panier protein, and we perform, painfully, I have to say, the antibody stainings on those developing elytra. And what we found is that, I hope you can see, there's not enough contrast, but I hope you can appreciate that basically in those developing elytra, in the different morphs, Panier is expressed wherever black pigment will appear. And conversely, Panier is repressed where red pigment will appear. It's mostly obvious, for instance, in this morph. Panier is expressed everywhere except here. In these guys as well, you can see that Panier is on pretty much everywhere except in the presumptive red dot areas. And it's the opposite pattern, basically, in this red morph. Panier is expressed where the black dots will appear in the adult. So we could put together the fact that the GWAS indicated that action is taking place at Panier. The Panier function is absolutely necessary to make black pigment, and there is a correlation between changes in the distribution of Panier and the distribution of where the black pigment would form. So we put altogether a model in which we propose that regulatory variation at the Panier locus is responsible for driving those different expression patterns of Panier, and that together are also responsible for the variation within the species. Now, we published this paper a few years ago, and at the same time, another group led by Teruyuki Nimi in Japan basically came up with exactly the same conclusion using slightly different methods. So it was very reassuring to find another lab finding exactly the same thing, and luckily at the same time, so there was no scooping. So now, Panier is responsible for this diversification, but then we can wonder how come we find all these morphs in nature? Because in fact, we can find them all in the same location, they mate just fine, so we might wonder how come those discrete forms are maintained in nature. Well, when we assemble the genome of the different morphs, what we found is that in fact, as you would expect from individuals belonging to the same species, you have a pretty good alignment along the length of the genome, and this is focusing on the Panier gene, except exactly at the Panier gene, exactly at the region where we found all those SNPs accumulating and distinguishing the red from the black morphs. And if I zoom in on what we found, it's here. Basically what we found a footprint of an inversion, basically, that is spanning about 50 kb within the first intron of the Panier gene. When we compare the genome assemblies of the different morphs, not just these two, but the four morphs that we had sequenced, we found, in fact, a very complex pattern of multiple inversions distinguishing the different morphs. And we think that those inversions are preventing recombination, as has been discussed yesterday, and that those inversions, helps to maintain the discrete morphs in the natural population. Now, it's also been known from the literature that the interaction between alleles also contributes to the diversification of coloration pattern, and this is shown in this cross scheme. If you take any of those morphs and you cross them to their counterparts, you see that the product of their offspring, basically the F1, has a pattern that is different in most cases from any of the two parents. And this has been noticed by Tan more than 70 years ago and has been named mosaic dominance, because what happened is that when you look at the hybrids, you see black pigments wherever there was black pigments in either of the two parents. And now it makes complete sense if you think about the role of Panier that I've described to you before. You can see how if you combine basically those two alleles in these F1 hybrids, you will have a superimposition of Panier expression everywhere in the elytra where Panier is expressed in one of the two parents. And you can see, for instance, that in this area here, there is Panier expression being expressed all over the elytra, more or less. Here it's excluded, except in this little indentation here that comes from the red morph allele. And that gives rise to this black indentation in the red dots in the F1 individuals. Okay, so far what I've shown you is that the diversity of coloration pattern in Harmonia axyridis is fueled by allelic diversity at the Panier locus, by interaction between the alleles. But as I told you in introduction, we also know that developmental plasticity can contribute to some extent to this diversification of this pattern, and I will show you that in the next slide. So this is what happens if you raise Harmonia axyridis, the red morph or any of the black morph, I've shown you only one here, at different temperatures, 18 degrees, 25 or 29 degrees. What you can see with the naked eye, of course, for the red morph is that there is a very strong response when it comes to the amount of black pigment that is deposited and the pattern on the elytra and also in the thorax, actually. You can see that basically if you raise the flies, sorry, not the flies, the beetles at 18 degrees, I rarely talk about that. At 18 degrees, they have much darker than at 25 or even at 29. Basically, there are almost no black pigment appearing. You see those two small dots remaining. In the black morph, the situation is a bit less obvious. With the naked eye, you may not see any difference, but if you look closely, and we are just starting to do that, we can measure some differences. So this is a very preliminary result. I got the slide yesterday evening from my collaborator. If you just make a transect along the length, basically, of the elytra, focusing on the intensity of the black area, so the red dots here are just pointers to orient you within the elytra, but we're looking at the value, the gray value here, corresponding to this area here in between the red dots. What you can see is that the value that you get when you raise these black morphs at 18 degrees, this value is much lower than when you raise it at 29 degrees. And here it's a bit counterintuitive because lower value means darker than when the value is higher. So even though we can't really see with the naked eye the difference in the darkness intensity, we can measure it nonetheless. So there is what we call a low plasticity, fairly low plasticity when it comes to pigmentation intensity variation in the black morph, compared to a much higher plasticity in the red morph. And we thought that it was an interesting situation because obviously we know a lot about, and we know many traits that are developmentally plastic. I'm sure Patricia will talk about that in the next talk, of course. We know many genes that have been shown to contribute to some degree to some plasticity for many traits and many species. But I think we know cases, or I'm not aware of any cases, where we know the genetic basis of developmental plasticity variation. What makes a species or a population plastic for a given trait, as opposed to another species or population not or less plastic in response to the same stimulus. So we thought that this contrast between high plasticity and low plasticity in this species would be a good system to address this question. What distinguishes those two morphs? Well, again, plasticity, we've discussed that before. It's the fact that given a given genotype raised into two different environments will give two different phenotypes. Those two phenotypes can be fairly similar, although different. Well, I will call that low plasticity. Or instead, it could be they could be very, very different from one another, leading what I call a high plasticity. And we can see very basically that the genetic pathway that will be responsible for the expression of a given phenotype can be modulated by plasticity module. I will englobe that very roughly. And then we can wonder where would be the variation responsible, the genetic variation responsible for the variation in plasticity. And we can wonder to what extent the genes that contribute to the variation in the trait formation are also responsible for the variation in trait plasticity. So this is illustrated with these two schematic examples. You can imagine that it could be variation in the plasticity module itself. That would be, let's say, more efficient or stronger in one genotype compared to another one. Or inversely, we can imagine that the plasticity module is basically the same in those two genotypes, but the ability to respond to the output of the plasticity module will be different between those two genotypes. And so we thought we could try to address this question again with Harmonia. So we went back to a mapping approach, looking for basically SNPs accumulating and distinguishing between the red highly plastic morph and the black lowly plastic morphs. In addition to that, we wanted to, if we find candidate genes, identify genes that would show some kind of temperature-dependent variation, either in activity or in their expression. So again, we repeated, well, again, let's say we, it's Mathieu and Arnaud repeated their GWAS analysis using several populations of high plasticity red morphs and low plasticity using a new genome-wide quality assembly. They identified 1,300 SNPs, which when you assemble into some fancy methods, allow to identify 8 candidate genes. And again, just to give you a pointer, this big, this time blue tower is Panier. But we also found some other candidate genes that we neglected in the previous study. So, as I said, then we wanted to, in addition to these candidates, we wanted to find a way to sieve through those candidates and try to connect any of them, ideally, with variation in response to temperature. Of course, assessing activity for some of these genes is very difficult, so we turned to an easier experiment, making the hypothesis that the expression level may vary with temperature in the developing elytra. So again, we compared through RNA-seq the response to temperature during elytra formation in the red morph, contrasting 18 versus 29 degrees, the black morph, contrasting 18 to 29 degrees, and the F1 hybrid, contrasting 18 to 29 degrees. And this is the result. And I won't go through details about this plot. Basically, it's our typical volcano plot where you can compare gene expression. And basically, to cut a long story short, when we look at how the different candidates we found from the GWAS analysis that I was showing you just before are distributed, we found that basically only one passed the threshold that we've set, which is Panier. So Panier is differentially expressed with temperature in all the 3 conditions. And it's the only of the candidate gene that show this behavior in at least at the stage and during elytra formation. Now, importantly, even though Panier responds to temperature in the 3 genotypes, it responds to a different degree. So the magnitude of fold change is different between the morphs. And this is shown in this graph, so I will walk you through it. So these are the different genes, 73 genes, that we found differentially expressed depending on temperature in the 3 conditions, the red morph, the black morph, and the F1 hybrids. And what we're looking at here on this graph is the percentage of increase in fold change when we compare 18 and 29 degrees between the black morph, the red morph, and the hybrids relative to the black morph. So everything is normalized to the response to the black morph, just to show, to polarize the changes. And what you can see is that in the red morph specifically, there is a much stronger response to temperature of Panier. Actually, Panier is one of the most responsive genes in this morph. So this is good. Well, this is interesting, I should say. We know that we have SNPs accumulating around Panier that correlates with the changes in the intensity of plasticity response. We see a difference in expression level in response to temperature between the red and the black morphs, but this could be just due to background effect. So to distinguish between background effect and regulatory variation between those 2 genes, or between those 2 alleles, sorry, the red and the black alleles, we exploited the F1 hybrids that I've shown you before. And we made the hypothesis that when we make those hybrids, between the red and the black, which is this is the pattern we get. And when we raise those hybrids at different temperature, the result is shown here on the left-hand side. You see that there is a bit of variation, but not so much. It's mostly visible at the level of this indentation. And we make the hypothesis that that would correspond to a decrease in expression of the red Panier allele specifically, that would be therefore more responsive to temperature than the black Panier allele. So we tested these experiments. Actually, this is an experiment I've been dreaming of doing for about 20 years, inspired by the power sequencing experiment that Trisha pioneered about 20 years ago. It's funny because we discussed about that at lunch break. So I've been keeping this idea in mind for a long time. Anyway, so the notion is that when you make hybrids, you can, if you have SNPs that will distinguish the coding sequence of the two alleles, you can quantify very precisely within the hybrids, at the same developmental stage, within the same genetic background, how many transcripts come from, in that case, the red alleles, and how many comes from the red alleles, right? And here, of course, you would only measure differences that are due to cis-relatory variation. And we can do the mapping, and we can do, therefore, the counting of the reads, and measuring, therefore, proportions of reads coming from the red or the black allele. So we did these experiments, and with the idea that if there is a different response to temperature between the two alleles, we should have a different profile. So this is what we expect to see in terms of proportion of reads coming from the Panier allele. We could have done the same thing with the black allele, doesn't really matter, coming from the red allele, if there is no response to temperature between the two morphs. We would expect that this proportion, whatever the value, would not change at the different temperature. Conversely, if there was a different response to temperature of the two alleles, we would expect to see that there might be a slope, a reaction norm, either positive or negative, doesn't really matter, this is theoretical, that would inform us that indeed the two alleles are responding differently to temperature. And if I'm showing you this, is because that's exactly what we found. When we look at the 4 SNPs that distinguish the black and the red alleles, which correspond to these four blots here, we found that indeed there is a decrease in the proportion of the red alleles. So that means that even though the red allele is expressed at a lower level, it has a stronger response to temperature than the black allele when they are in the same environment. So the red allele from Panier seems to be more sensitive to temperature variation than the black allele. So we found that in this case, cis-relatory variation at the Panier gene determines both differences in color patterns, the mean value of these color patterns, so basically distinguishing what happens in this morph from what happened in this morph, but also the ability of these morphs to respond to temperature variation. Now, to finish, I would like to open with something because of the theme of the conference, because I didn't pronounce the word ecology so far, because I don't know nothing about ecology, but I would like to point one thing that is, I think, very intriguing observation. Harmonia axyridis has been used throughout the 20th century as a bioagent to control aphids that are so dear to David, that he mentioned this morning. Go back, you know. And, well, the thing is, they've been used since 1916, first introduction in the US. And they've been used in the northern US, northern America, and Europe. And they were reintroduced every year because they would not overwinter, right? So there was not a problem. They would just need to import new individuals. They all come from Central Asia or East Asia, mostly China. And they were imported basically every year to kill the aphids. Fine. But then what happened in the late 1980s in the US, people started to notice that individuals managed to overwinter. They managed to acclimatize. And that was the starting point of a very successful invasion. This species, Harmonia axyridis, has now invaded basically almost all parts of the world in just 30 years. And I bring this up because the different morphs, when they were imported, people didn't pay attention to which morph they picked. They picked individuals, ship them to US or Europe, and regardless of the color. But what is really remarkable is that the only color morph that have been able to invade other area is the red morph, the one that is plastic to temperature. So I'm just raising it up because we have no data to support anything, but we suspect or we submit that the thermoplasticity of the red Panier allele in the red morph may have contributed to the invasion success of Harmonia axyridis. And this is something we would like to investigate in the future if my collaborators are still willing to work with me. So I will thank end by thanking the people who did all this work. So from my lab, it was the postdoc Junichi Yamaguchi, and as I mentioned before, from the CBGP, Mathieu Gauthier and Arnaud Estou. And I thank you very much for your attention.